Molecular biology is situated at the intersection of biochemistry and genetics. As these scientific disciplines emerged and developed in the 20th century, it became evident that they both aimed to determine the molecular mechanisms underlying vital cellular functions. The advancements in molecular biology have been closely tied to the creation of new technologies and the optimization of those technologies. Numerous scientists have contributed to the advancement of molecular biology. Therefore, the history of the field depends on an appreciation of these scientists and their investigations. An attempt to comprehend the molecular mechanisms of genetic inheritance and the structure of a gene gave rise to the field of genetics. Welcome back to my channel. In 1866, Gregor Mendel, a pioneer in this field, first published the laws of genetic inheritance, based on his research into the hybridization of pea plants. One such law of genetic inheritance is the law of segregation, which states that diploid individuals with two alleles for a specific gene will only transmit one of these alleles to their progeny. The study of genetic inheritance is commonly referred to as Mendelian genetics, due to his influential contributions. The discovery of DNA structure was a significant turning point in molecular biology. Friedrich Miescher, a Swiss biochemist, began this work in 1869 when he proposed a structure dubbed nuclein, that is now known to be deoxyribonucleic acid, or DNA. By analyzing the components of pus-filled bandages and observing the unique properties of the phosphorus-containing substances, he discovered this unique substance. Phoebus Levene, who proposed the polynucleotide model of DNA in 1919, as a consequence of his biochemical experiments on yeast, was another notable contributor to the DNA model. Erwin Chargaff elaborated on the work of his predecessors, and elucidated several essential properties of nucleic acids in 1950. The sequence of nucleic acids differs between species. The total concentrations of purines, adenine and guanine, and pyrimidines, cysteine and thymine, are always equivalent. Currently this is known as Chargaff's norm. James Watson and Francis Crick published the double helix structure of DNA in 1953, using the work of Rosalind Franklin and Maurice Wilkins, in X-ray crystallography. Watson and Crick described the structure of DNA, and speculated on the implications of this unique structure for potential DNA replication mechanisms. Molecular biology delve into the inner workings of cells, and learn about the chemical processes that govern all living things. Discover how cells replicate, communicate, and function, and explore the latest breakthroughs in gene editing and personalized medicine. Molecular biology is the study of the chemical processes that govern all living things. From the tiniest bacteria to the largest mammal, every living organism is made up of cells, each with its own unique structure and function. Discovering the structures of biological molecules, as well as their interactions and how these interactions explain the findings of classical biology, was how molecular biology was initially defined, as an approach focusing on the underlying causes of biological events. Molecular biology is an incredibly exciting field, with new breakthroughs happening all the time. One of the most exciting areas of research right now is gene editing and personalized medicine. Molecular biology is not limited to the study of biological molecules and their interactions. It is a compilation of techniques developed since the field's inception that have allowed scientists to gain knowledge of molecular processes. In this manner, it has both improved and supplemented biochemistry and genetics as methods for comprehending nature that existed prior to its invention. The polymerase chain reaction, or PCR, which was invented in 1983, is a significant technique that has revolutionized the field. The polymerase chain reaction is a reaction that amplifies minuscule amounts of DNA, and it has a wide range of scientific applications. The central dogma of molecular biology defines the process of DNA transcription into RNA, followed by protein translation. The understanding of structures, functions, and internal controls within individual cells which can be used to efficiently target novel drugs, diagnose disease, and better comprehend cell physiology, is also dependent on molecular biology. The use of molecular biology, or molecular cell biology in medicine, is now known as molecular medicine. CRISPR-Cas9 is a revolutionary gene editing tool that allows scientists to cut and edit DNA with incredible precision. This technology has the potential to cure genetic diseases and revolutionize medicine as we know it. Personalized medicine is another area of research that has enormous potential. By analyzing a patient's genetic makeup, we can tailor treatments to their specific needs, increasing the chances of success and reducing the risk of side effects. Personalized medicine is already being used to treat cancer and other diseases, and researchers are continuing to make breakthroughs in this field. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats, which are a series of repeating DNA sequences that are found in bacteria and archaea. These sequences are believed to play a role in defending these organisms against viruses and other foreign invaders. Cas9 is a protein that acts as a pair of molecular scissors that can cut DNA at specific locations. By combining CRISPR with Cas9, scientists can program the system to target and cut specific DNA sequences within a genome, allowing them to add, remove, or replace genes with incredible precision. The potential applications of CRISPR-Cas9 are enormous. It could be used to cure genetic diseases, create new vaccines, or even edit the genes of plants to make them more resistant to pests and disease. However, there are also concerns about the ethical implications of gene editing and the potential risks of making irreversible changes to the genetic makeup of living organisms. Despite these concerns, CRISPR-Cas9 has already revolutionized the field of molecular biology and has the potential to transform many aspects of our lives in the years to come. Another incredible aspect of molecular biology is the way cells communicate with each other. Through a complex network of chemical signals, cells are able to coordinate their activities and respond to changes in their environment. In the human body, the nervous system is responsible for much of this communication. Neurons in the brain use electrical and chemical signals to communicate with each other, allowing us to think, feel, and interact with the world around us. As our understanding of molecular biology continues to grow, we're sure to discover even more incredible insights into the inner workings of the cells that make up all living things. It is common for researchers to combine molecular biology techniques with genetics and biochemistry techniques. The majority of molecular biology is quantitative, and a substantial quantity of recent work has utilized computer science techniques, such as bioinformatics and computational biology. Since the early 2000s, molecular genetics, the study of gene structure and function, has been one of the most prominent subfields of molecular biology. By studying the interactions of molecules in their own right, as in cell biology and developmental biology, or by using molecular techniques to infer historical characteristics of populations or species, as in fields of evolutionary biology, such as population genetics and phylogenetics, molecular biology informs other branches of biology. Biophysics also has a lengthy history of studying biomolecules, from the ground up, or molecularly. Molecular cloning is used since 1960s. Technique is to isolate and then transfer a DNA sequence of interest. A variety of systems, including inducible promoters and specific cell signaling factors, are available to facilitate the expression of the target protein at high levels. The protein can then be extracted in large quantities from the bacterial or eukaryotic cell. Then it can be examined for its enzymatic activity under a variety of conditions. Its tertiary structure can be crystallized, and in the pharmaceutical industry, the activity of new medications against the protein can be examined. Who knows what breakthroughs the future holds? Thanks for watching.